Depicted figure shows option 1 Munsell system, 2 CIE LAB system, or 3 3D chromatism, or 4 saturation system. So this question and this color system is extremely important. Now this has a single answer which is very evident that this is the Munsell system. Alright, so now let me explain you what the Munsell system is actually. So Munsell system and the CIEB lab system, which is the second option, these are the few color systems that we use in dentistry. So a color system is basically, or a color is basically made up of three components: value, hue, and chroma. So any color has a hue. What is hue? Hue is the actual color. For example, how you see here, this is blue color, this is green color, yellow, we have red, we have pink. So this is the hue of any color. So it is the basic color that you're picking up. Then the value. Value is the lightness or the brightness of any color. So this axis that you see in the center is your value axis. Okay. So it starts basically from 0. 0 is basically the darkest color, that is black. And as you keep on increasing, your color gets brighter or whiter. So the least value is the darkest, higher the value, brighter the color. Alright. Then the third thing is the chroma. Now chroma is the saturation or the intensity of the color. This gradient that you see, which is going from the center towards the periphery. So the color is the lightest towards the center and it goes darker as you go towards the periphery. So the Munsell system is based on these values, these proportions, basically the value, the hue, and the chroma. So these are the three important things that you need to remember. Now the second system that they've mentioned here is the CIE LAB system. This is a similar system to Munsell, but there are certain differences which we need to know because you may be asked about this as well. So the CIE part, this is an abbreviation to the International Commission on Illumination. Okay, that is known as CIE. Now the remaining part of the name, which is LAB. L basically stands for, these are the names of the axes that are there in this. So this is your L axis, this is your A axis, and this is your B axis. So basically this system has four normal colors, or the four normal colors that we see in our day-to-day lives: the green, blue, red, and yellow. Okay, so the A plus axis demarcates the red color, A minus axis is for green, B minus is for blue, and B plus is for yellow. You don't really need to memorize this, but you just have to understand. Like for example, if they give you this picture or something similar where you see the L axis, A axis, and B axis, then you should know that this is the CIE LAB system and not the Munsell system. Alright, so it depends on which all axes are there in the picture. So here, this is what you just need to understand, something similar to the Munsell system. Your L axis again starts from 0 and it goes upwards. The color gets darker. 0 is black. As you move towards the top, the color gets whiter. Then the chroma again gets increased from the center of the axis to the outer periphery. The intensity of the color on the periphery is darker. And these are the hues along the periphery that you see. They just have the axes A plus, A minus, B plus, B minus, that you need to just understand this system. Alright. Now the other two options that they've given, that is 3D chromatism, is mostly used for 3D printing. Not much of a color system that you need to understand. And the fourth one is the saturation system. It's just a name that they've given in one of the options. I hope this is clear. Apart from these two systems, you may also be asked that which is the important part of or component of color which matters the most during shade selection. So it is the value of any color that matters the most for us during shade selection in dentistry. So just remember, value is very important. Okay, thank you.